remember the defining feature is that the last three carbons, you write them like glycerol, replace one of the OH, the middle OH with NH2, okay? And then there is this very small molecular formula on the first carbon that gives you an idea, or that will help you remember what sphingosine is. Then if I give you a structure like this and ask you what this is, it should be very easy for you to recall that this side is a sphingosine derivative, this is glucose, so it has to be a glucocerebroside. Okay? Again, look for common motifs being repeated in the lipid structures. If you do that, it will be very easy for you to remember and recall any kind of lipid structure. And again, don't bother with with writing CH3, CH2, CH3 again and again and again. Okay? In chemistry, if you have a structure like this, each point is a carbon. Okay? It's a given. And if nothing else is written, then all these carbons are being, all the bonds are being saturated by hydrogen. Okay? Okay. Now, coming on to the fused ring structure. Till now, we were talking about the open chain fatty acids or the open chain lipids. Now we are looking into the structure of the fused chain lipids. And it's a collection, it's a huge collection of different compounds. The main defining feature is that it's a mixture, or no, it's a structure, that consists of 4 rings. Okay, there are 3 rings with 6 carbons and 1 ring with 5 carbons. Okay, again, this is here for you to remember what a steroid looks like. Okay, if I ask you give you a structure and ask you what is this, if I'm asking about a steroid, look for this defining structure. Okay, there may be a bond missing here or there, but if you have 3 rings, okay, with 6 carbons and each, and 1 ring with 5 carbons, okay, arranged like this. The A, B, and C rings are the 6 carbon rings, and the D ring is the 5 carbon ring. This is your basic steroid structure. Okay. The most popular example is cholesterol. Okay. Again, if I just ask what is this, look for this structure. If you write steroid, most of the times you can get away with the correct answer. Okay. Remember, with respect to cholesterol, there is a hydrocarbon side chain, okay, with 1, 2, 3, 4, 5, 6, 7, and 8. And 8 hydrocarbon chain attached to the D ring, you get cholesterol. Okay. Again, any confusions about what a fused ring structure is? Okay. Again, there are a number of fused ring structures. Only and only if you have 3 6 carbon rings and 1 5 carbon ring fused in this fashion. That's the only point where you call this structure a steroid. Okay. Again, today we are just looking at the structures, or how to basically identify lipids. Okay. We're not really going into their roles or biological roles. Now, the important physiological aspect of cholesterol, besides cholesterol being an important, a very important component of the cell membranes. Okay? Cholesterol is the structure, as you can see with the ring, okay, of cholesterol. It doesn't have the same freedom to move around as a long chain, long hydrocarbon chain. It is a very rigid ring structure. Okay? And the rigidity of the cell membranes in most of the plants and animals is due to the presence of cholesterol. Okay? And that is why cholesterol is not always a very bad thing. Cholesterol in very high amounts is a very bad thing, but it is an absolute requirement for any kind of living system. There are no known mutants for any kind of plant or animals that can survive without cholesterol. Okay? If there is a plant or an animal with like a very extreme mutation which would result in no cholesterol being produced, that animal or plant cell does not survive. Like there is no way, there is no replacement for cholesterol, okay, in the biological system. So again, this fused ring structure makes cholesterol a very rigid structure, and that is why when cholesterol is introduced into the cell membrane or the membrane bilayer, the bilayer becomes very rigid in structure. Okay, it changes the melting temperature of that plant membrane or that cell membrane. I'm sorry. Now, the other important aspect wherein cholesterol plays a role are the sex hormones in animals. Okay, so progesterone, testosterone, and estrogen. Okay, progesterone is, what do you call it? Is a molecule from which both testosterone and estrogen can be derived. It is common in both females and males. Testosterone is the male sex hormone, estrogen is the female sex hormone, but again, you have different ratios of both in both males and females. Again, if you just look at the structure, all these sex hormones are derived from cholesterol. That's why cholesterol is so important. Okay. Now, coming on to the last portion of the derivatives of, again, and there is not too much detail in it. Again, I'm not going to really bother you with remembering these structures, but the important lipids, the other aspect of fused ring structures, or even open chain structures. Again, vitamins are a mixture of both open chain and fused chain structures, but there is usually a ring structure present. Okay, and again, there can be no amount of words that can be used to describe the importance of vitamins in our life. Okay, the first example is vitamin A, and it has a very crucial role in providing us with proper vision. Okay, the compound from which vitamin A is derived is beta carotene. Okay, name suggests, derived from carrots. All of us with glasses never ate carrots growing up. So that was not the case. But so beta carotene, when it gets cleaved right in the middle, okay, you end up with retinol. Okay, retinol is directly used in the retinal cells. Okay, or you have, if you have an aldehyde group being attached to the, or aldehyde group replacing the hydroxyl group, you end up with retinol, which is the aldehyde derivative of retinol, and it is retinaldehyde, which is actually your vitamin A, which is used by the retinal cells and gives you perfect vision. Then you have vitamin D. Like I said, look for the ring structure. There can be a few bonds missing here or there. Prime example is vitamin D. The ring structure is not closed. There is a double bond this end. But then, if you just look at it, 6 carbons here also, another 6 carbon, and a 5 carbon ring. Okay, so that's vitamin D, and vitamin D is very important and is required for both calcium and phosphorus metabolism within our bodies. Okay, vitamin E in hair, everything. Okay, doesn't let you age. That's vitamin E. Okay, again, vitamin E is very similar in structure to vitamin A, except that vitamin A has a single ring structure. Vitamin E has 2 ring structures. Okay, what makes vitamin E very important with respect to aging, or as you are aging, you should be taking more and more vitamin E, is that it's a very important antioxidant. Okay, it's a very important antioxidant system, which basically scavenges all the reactive oxygen species within the cell, which can oxidize your proteins, which can oxidize your cell membranes, leading to premature cell death. Vitamin E helps scavenge those, and that is why it is so important. And then you have vitamin K. Again, vitamin K looks very similar to vitamin E in its structure, except for the presence of 2 double bonded oxygens in the second ring, and the hydrocarbon chain that is attached to one of the rings can be as long or as short. Okay, But this basic structure basically gives you vitamin K1 and K2. And the vitamin K, the word K comes from Danish. That is coagulation. Okay? They just decided to write coagulation with a K. Okay? And that's why vitamin K. The name coagulation, that they were talking about blood clotting. And it's a very important factor which is required for blood clotting. And that is why vitamin K is so important. Now, the important point that you should remember about vitamins is that they are required in very small quantities. Okay? Don't let the people at Walgreens fool you about, having too many vitamins. Okay? As they say that, Americans are just pissing vitamins, and that's the truth. Okay? So they are required in very small quantities by the system, but they are extremely essential in their function, and they are an absolute requirement for most of the functions like blood clotting, calcium metabolism, okay, vision. Now, this was just the basic introduction to lipids, and basically what we have done today is that we have covered the portion for 2 classes, but then, I don't really see any more lipid function. Now, the other thing that this chapter also includes is the function of cell membranes, but I like to teach cell membrane at the time when we are discussing transporters and receptors with respect to proteins. Okay? That's the point, that's the time when I bring in the lipid bilayer structure also, because then it gives you a refresher with respect to the lipids. What is the structure of lipids? What is the functionality of the lipids with respect to their structure? How they form bilayers, and when they interact with proteins, what is the function of those proteins in the bilayers? Okay? So, if we do not reach to the cell membrane part in this portion before your exam, this is what you need to be concerned with respect to lipids. Okay? Now, let me remind you again, Tuesday is your quiz. If you do not feel the need to be here on Tuesday, free to do so. It's going to be a 20-point quiz. And any and all of those 20 points, depending on what you decide to make on that quiz, will be added to your first exam.
You only have 200, right? You only have 200.
Yes, very important point. Thank you. Only up to 100. Okay? You will not get any points over 100 on your first exam. Yes.
So you wanted to know the names and structures of all of the lipids we covered except for the vitamins?
Yeah, for the vitamins, do not bother with the structures. Certainly, most of you do not. But with the lipids, whatever you have here, not the whole structure, but what defines that lipid. Okay, if I ask you what is phosphatidylcholine, okay, the name phosphatidyl, that's very simple. Okay, that's phosphatidic acid. Okay, and choline, you should be able to recognize that from a mixture of structures. Yeah, if you want to use it through what? Okay, another important point. That these quiz points, okay, these are freestanding points, okay. I will not straight away attach them to your first exam, okay. Freestanding points in the sense, if you want to use them later also, you can do that. Oh, okay. Yes.
I didn't know.
That's the point. I mean, the idea is not to be unfair with anyone. If you've already made an A, you have like 95, 98. What's the point of turning up for the quiz and making 2 points on that quiz, getting 100? So, and also it makes it makes sure for me that you will turn up. Those 20 points are there for you to use.